Okay, so welcome back to返學 DSE. So we're going to talk about chapter 15 about detection. So for detection, we have three different components. There is stimulus, effector, and also response. So the stimulus will trigger effector, and the effector will trigger response. So the first question is, how can we pick up the stimulus? So there will be generally four types of receptors: photoreceptors, mechanical receptors, chemoreceptors, and also thermoreceptors, and they are responsible for different situations. And first, we will focus on the eyes. So in our eyes, we have a number of structures like eyebrows, eyelash, and also the eyelids. And each of them will have their own functions. If you can remember them, please do so. If you can't, just leave it, because I know that there are so many things in biology you are required to recite. And there are also tear gland. Tear gland will produce tear, and you are kindly reminded that in the tear, it contains an enzyme called the lysosome, which can kill bacteria. That's why tear is also involved in the immune system. And then for the outermost layers of eye, there's Cascaria, and it is a tough layer, and it is to protect the inner structures and to maintain the shape of the eyeball. And for the outer part, it also contains the cornea, and cornea is protected by the conjunctiva. And they both serve the function to reflect light. Remember, refract this in the middle is RO, not LO. Okay, this is very important because refractions mean the lights are not bouncing back, but they are converging to somewhere. And there is also a contour, and then it contains a back pigment to reduce the reflection. Remember, this is the FLO, not FRO. Okay, very important, the spelling, because for the one with the LO, it means that the light is bouncing back, so you reduce the chance of light bouncing back and forth inside your eyeball. And then there is an iris. Iris is to control the size of the pupil to determine the amount of light entering the eye. And for the retina, it contains two types of photoreceptor. They are called the rod cell and cone cell. And for the yellow spot, it is concentrated by cone cell. And there is a place of attachment of your optic nerve. That place is also known as the blind spot because the optic nerve is attached there. That's why no photoreceptor is on that spot. And then the ciliated body. The ciliated body is connected to the lens by the suspensory ligament, and which contains the ciliated muscle. So the contract and the relax of this muscle will change the thickness of the lens. And then now becomes the lens. The lens is a transparent and elastic structures, and it is to refract light. Okay, refract is mean either diverging or converging the light and to focus the light onto the retina. So the relationship will be the ciliated bodies control the suspensory ligament, and then ultimately it controls the thickness of the lens. So there will be two types of humors. For the first one is the aqueous humors, is in the front part of your eye and is to maintain the shape. And similarly, it is another type of humors, aqueous humors, and also it is to maintain the shape of your eyeball. And this is how we see. When lights come into the eyes, the light ray will be refracted and focused on the retina. The image will be viewed upside down, laterally inverted, and also smaller than object. And then the image is detected by the photoreceptor, that is the light-sensitive cell, and then it will send the nerve impulse back to the brain. And then the nerve impulse once transmitted to the visual center, and then it will be interpreted as upright image. And for the light-sensitive cell, let's talk about rod first. For the rod, it is responsible for the vision in dim light, and it is responsible for black and white vision. And it distributes throughout your retina, except two places, that is the yellow spot and also blind spot. And reverse, we talk about cone cell. So the cone cells work best in black light and is responsible for color vision. And we have generally three types of them, that is red, green, and blue. And for the cone cell, it is concentrated at the yellow spot as previously discussed. And for the blind spot, as I told you, there is a place that no photoreceptor can be found. That's why when an image formed there, you cannot see it normally. But however, we don't normally notice in our daily life. So what is the reason behind? In your lesson time, your teacher should have done the demonstration and also the experiment with you guys. In order to control the amount of light entering the cell, we have to change the size of the pupil. These two terms you have to bear in mind, especially for the English group, because the term constrict and dilate are restricted to this kind of usage. Because this is not a muscle, pupil is not muscle. That's why you cannot say relax and contract. You can only say constricts and dilates. So here is the general phenomenon, and I would suggest you just remember one cases instead of two cases. Let's talk about the one in the bright light. In the bright light, the circular muscle will contract and the radial muscle will relax, and in return, the pupil will be constricted, and then the amount of light entering the eye will be decreased. And as we are focusing for the near and distant object, we are going to adjust the thickness of the lens. So let's focus on the near object. For the near object, the circular ciliated muscle will contract and then the suspensory ligament will strengthen. I will recommend you to remember the term tension down, okay? And with a less tension in the ligament, the lens will become more convex, okay? And then the light refraction will be increased. That's why the bending is also increased. The distant object will be blurred because the image are usually formed in front of your retina. So there are two causes of it. Either the lens is too thick or the eyeball is too long, and the correction will be wearing concave lens. And long-sighted will be a reverse case. For long-sighted person, distant objects are clear, but close objects are blurred, and the image are formed behind the retina. And the case and also the correction will be a reverse of the short-sighted. So I recommend you to remember the case short-sighted. So the color blindness, it is a defection of the cone cells because it is responsible for the color vision. And usually there will be two types, either the red-green color blindness or total color blindness. And the causes usually are genetically inherited. So that's why, in general, the correction will be no way. However, we are also finding ways to solve this problem. After talking about the eye, we will move to the concept about ear. In ear, there are even more terms. To me, I recommend you to focus on important terms. In here, eardrum. Eardrum is the membrane that vibrates so that the sound waves are reached. Okay, the term vibrate is very important because our hearing is simply based on vibration. And then the ear bones will amplify and transmit the vibrations from the drum to the over windows and then later on to your inner ear. And for this part, I will recommend you to external tubes. This is to equalize the pressure and explain why we have a bad feeling or painful feeling as the flights are taking off from the ground or either the flight is landing. And then the semicircular canal is also recommended in your revision because it is helping you to keep your body balance. And for the Korea, I will not recommend you to remember these three terms, but sensory hair cell, I don't think is a very difficult term. You are kindly reminded that it is the final steps that is connected before your nerves. That's why when you stimulate the sensory hair cell, you are generating your hearing. And here is the process of hearing. So it is quite a long step and quite a number of terms there. Remember, the important concept is the vibration and also amplification by the ear bones. After talking about the ear, we will move to plants. For plants, there will be three types of general stimulus they would like to pick up. There's the light, water, and also gravity. However, the steps are resulting in slow response, and this response we will group it as tropism. And the main focus of our syllabus will be phototropism. For the shoot and roots, they will respond differently to the light. For the shoot, it will grow towards the light, while for the roots, it will grow away from the light. And here we provide an explanation why this phenomenon will help the growth of the plants. And then we will use cellulose in our studies. Because of these reasons, it is easy to observe, it grows rapidly, and also it is small and easy to handle. And we will talk about the story of Auxin. For the Auxin, there are some chemical produced in the tube that is the shoot part, and this chemical will move down by diffusion, and it may cause or may result in an uneven distribution of this chemical because of light, and then the higher concentration will lead to a faster growth. This is the general story behind. So the specific term of those chemical will be auxin, and we are usually linked with the nature of science. We're asking students how they are going to make their hypothesis and how they are going to relay their observation and inferring. And the most common type of auxin will be IAA, and it is produced in the apical meristem, which we talk about in chapter 4. And then the effect is happen in the region behind it, that's the region of elongation. So remember of those experimental setup and design. And this is how auxin concentration will lead to the growth of the shoot and roots. So the low auxin concentration will promote the root growth, while the higher auxin concentration will promote the shoot growth but inhibit the root growth. And then I kindly and strongly recommend you guys to able to sketch related diagram. And here is the answer of it. So you may notice that for the root one, low concentration is the stimulation. However, in a higher concentration of the auxin, it is an inhibitory effect. And for the shoot, a higher concentration usually results in a stimulation. However, an extreme high concentration of auxin will still inhibit the growth. So the light, if it is a uniform light, then it will result in an even distribution of auxin. However, if it is a unilateral light, the shaded region will have a higher auxin concentration, and the higher auxin concentration will promote the shoot growth. That's why the shaded region will grow faster, and it will result in the shoot bending towards the light, and it will be the reverse case for the root. So here is the extract from the HKGA document. In here, you may notice that you have to understand major parts in eyes, and also rock cell and cone cell, how they are responsible to our colored and also black and white vision. So this is the color vision part. And for the eye accommodation is how we are adjusting our lens for a near and also a distant object, and how we are adjusting to the bright light and also dim light. And there will be long sight, short sight, and color blindness are the eye defects that we have to talk about. And for the light stimulus of the plant growth, that is tropism, we have to understand for the response of the fruit, roots and shoots are different, and the role of auxin in their growth. As for the hearing part, you are required to know some major parts of the ear. However, you may also notice that the mechanism of hearing is not the learning and assessment focus. When you are doing your revision, you should be selective. So this will be the end of the intensive video of chapter 15. So see you guys next time. Bye.